Good afternoon, and welcome to this webinar titled Screening Approaches for Reducing Attrition Due to Drug-Induced Liver Injury Early in Drug Discovery, brought to you by Xconomy and presented by Eurofins. My name is Maggie Whalen with Xconomy. Before we begin, let me explain how you can participate in today's presentation. First of all, if you have any technical difficulties during today's session, please press the Help button on your player console to receive assistance in solving common issues. We welcome your questions during today's event. In order to submit your questions to today's presenter, simply type your question into the question window on the left-hand side of your screen and then hit the Submit button. We'll be answering as many questions as possible during the Q&A session that will follow the main presentation, but please feel free to send in your questions at any time and we will add them to the queue. If at any time you are having audio difficulties or the advancing of the slides, simply hit your F5 to refresh your webcast console. Please also be aware that today's sessions are being recorded and will be available on the Xconomy website, xconomy.com, beginning tomorrow for you to review. You'll be notified by email when the archive is available. Let me now introduce today's presenter, Yang Zhao, PhD, Director of In Vitro Toxicology Services, Eurofins Discover. Yang, the floor is yours.
All right, thank you for the introduction. Thank you for attending my presentation. My name is Yang Zhao. I'm the Principal Scientist and the Study Director for In Vitro Toxicology Studies at Eurofins Discovery. I have been managing the In Vitro Toxicology Services at Eurofins Discovery for over 15 years. Today, I'm going to talk about the in vitro screening approaches for reducing attrition due to drug-induced liver injury, in short, DILI, early in drug discovery. And in particular, I will talk about what in vitro assays that we offer at Eurofins Discovery to de-risk your drug candidate's DILI liability. And this is the outline of my presentation today. First, I will talk about the background of DILI. Why is it important to assess DILI risk at the early stage of drug discovery? What are the causes of DILI, and what are the typical strategies that pharmaceutical companies utilize to assess DILI risk using in vitro assays? At Eurofins Discovery, we have developed and validated two panels of in vitro toxicity assays to predict DILI risk. And those are DILI screening panel and the DILI mechanistic panel. I will talk in details about those assays in the two panels and the utility of the panel in early drug discovery process. Hopefully, after my presentation, I will provide you with general information on what in vitro toxicology assays are important and useful to de-risk your drug candidate's DILI liability. At the end, I will give you a brief overview of admin toxicology services at Eurofins and leave about 10 to 15 minutes to answer any questions that you may have. First, let's ask a very important question. Why is DILI assessment in early drug discovery important? As we know that drugs are the major cause of liver injury. More than 900 drugs, toxins, herbs, and dietary supplements have been reported to cause liver injury. In the United States alone, approximately 2,000 cases of acute liver failure occur annually, and drugs account for over 50% of them. Approximately 75% of idiosyncratic drug reactions result in liver transplantation or death. DILI is also a major attrition for drug failure in preclinical and clinical trials, which results in tremendous loss of investment and time in drug discovery. DILI is the most common reason cited for withdrawal of an approved drug from market. An analysis done by the Organization of Drug Discovery data in 2018 found that among 192 drugs which were approved but subsequently withdrawn from the market worldwide, hepatotoxicity is the leading reason for attrition, followed by cardiotoxicity, and to a much lower extent, neurological, renal, and gastrointestinal related toxicity. A study published by the Tufts Center for Studies of Drug Development in May 2019 estimated the average cost to bring a drug to market is $2.6 billion and takes over 10 years. The cost of drug withdrawn from market is huge. Therefore, it is critical to assess DILI liability early in drug discovery in order to de-risk drug candidates and improve the success rate of drug discovery. Why the DILI risk of drugs that cause the DILI clinically was not discovered at the early drug discovery phases, as we know that the vast majority of toxicity data has been collected in animal models because they represent whole organism biology and there was limited availability of human models. But the animal models are limited in their ability to predict the potential for new drug candidates to cause DILI in humans. A recent pharmaceutical industry survey analyzed the proportion of positive preclinical liver toxicity findings that had positive clinical findings and found low concordance, showing human predictive value for DILI are only 33% by rats, 27% by dogs, and 50% by monkeys. Secondly, the animal experiments have the limitations of high cost, long study duration, and ethical concerns. The recent 3R movement called for replace, reduce, and refine the animal uses in toxicity testing. All of those reasons promoted the in vitro assays using cells originated from humans to the center stage of drug toxicity assessment in recent years. Most importantly, the recent development of in vitro technologies, especially in the use of human primary cells in toxicity testing, including 3D culture, microphysiological system, and tissue organ engineering, improves the in vitro testing productivity value and made the in vitro system made the in vitro assays more acceptable in the pharmaceutical and biotech drug discovery industry. So as shown in this drawing here, for majority of drugs, after they are administered to humans, they are absorbed to blood, accumulated and metabolized in liver, and then being cleared. When the drugs are processed by the liver, they have plenty of time and opportunities to cause liver injury. This explains why liver is the most vulnerable organ in human regarding drug toxicity. And clinically, drug-induced liver injury can be classified into intrinsic liver injury and idiosyncratic liver injury. Intrinsic drug reactions are caused by drug-related properties. Drugs that fall into this category cause reproducible injuries in animals, and the injury is dose-dependent. The injury can be due to the drug itself or to a metabolite of the drug. The daily dose and lipophilicity of a drug are found to be related to DILI. A research done by Chen in 2013 found that oral medications at daily doses of higher than 100 milligrams and a lipophilicity of log P greater than 3 to be significantly associated with severe DILI. This is called rule of 2. Most drugs are metabolized in liver, and some generate reactive metabolites. Those reactive metabolites can covalently bind proteins to generate drug protein adducts that might trigger immune-mediated reactions or cause direct toxicity. Formation of reactive oxygen species, ROS, can cause oxidative damage in liver. Increased ROS can directly damage DNA, proteins, enzymes, and lipids in cells and tissues, and induce immune-mediated liver damage. And DILI drugs can damage mitochondrial function by directly damaging mitochondrial components or by altering their metabolic pathways. Mitochondrial damage could trigger hepatic necrosis and or apoptosis, leading to activation of cell death signaling pathways. It has been reported that DILI drugs can impair mitochondria respiration and beta oxidation. trigger mitochondrial membrane disruption, and damage mitochondrial DNA. There is a significant association between loss of mitochondrial integrity and the potential to cause DILI. Inhibition of hepatic transporters, particularly the bile salt export pump, BSEP, could cause DILI. BSEP are responsible for the biliary excretion of bile acids. The inhibition of BSEP leads to the accumulation of cytotoxic bile acids in hepatocytes. Inhibition of other hepatic transporters, such as PGP, MRP2, 3, and 4, is reported to related to DILI risk. Recent research showed that the antagonists of a nuclear receptor FXR, which upregulates the expression of those transporters and downregulates the bile acid synthesis in hepatocytes, also contributed to DILI risk. Induction of steatosis and phospholipidosis are also related to DILI risk. Once cells are damaged by a drug, innate and adaptive immune responses play a significant role in driving tissue inflammation and injury. The second category of DILI is idiosyncratic DILI. Most idiosyncratic DILI cases happen at a therapeutic dose in an individual, even though this dose is well tolerated in the general population. Idiosyncratic DILI reactions are related to the characteristics of individual patients, including demographic factors such as age, gender as race, genotype, underlying disease, and co-medications. Idiosyncratic DILI is considered dose-independent. The assays that are currently available are relatively suited for assessing intrinsic DILI risk. It is very challenging, if not impossible, to predict idiosyncratic DILI by in vitro assays at current state. And because DILI can be a multi-step and multicellular disease process, the prediction of all forms of DILI is inherently impossible by a single assay. In a publication this year in Nature Review Drug Discovery, a group from European pharmaceutical companies and academia partners proposed a roadmap for the development and the deployment of in vitro predictive models for DILI. They take an approach contains a tiered system utilizing currently available in vitro assays and integrating with emerging cell-based technologies for DILI assessment. For example, the tier 1 in this roadmap comprises single-cell systems, such as using human primary hepatocyte cell lines, for instance, HepG2 cells, and reporter signaling. And those single-cell systems evaluate immediate chemical biological effects, such as bioactivation, drug or bile acid accumulation due to transporter inhibition, mitochondrial toxicity, and the signaling associated with oxidative stress, endoplasmic reticulum stress, and inflammatory signaling. And tier 2 assessment in this roadmap are more complex systems containing liver cells in a more physiological state, including assays in multi-cell culture, spheroid, and 3D culture, long-term culture, and microphysiological systems such as organ on a chip. The main goal of the tier 2 test system is to assess long-term effect of drug exposure. And tier 3 comprises a complex test system with the goal to be able to provide prediction on idiosyncratic DILI. And at European Discovery, we also take this tiered approach with the aim of managing DILI risk and assisting in the design and development of safer and more effective medicines. We employ a panel of assays to assess one or multiple factors that have indications to cause DILI. We offer 2 panels of in vitro assays in order to assess DILI risk at the early stage of the drug discovery process, the DILI screening panel and DILI mechanistic panel. The assays in DILI screening panel are high throughput and low-cost assays. They are suitable for large amounts of compound screening, such as compound library screening, to weed out the obvious offenders. There are 3 assays in this panel that assess the general cytotoxicity by running a 72-hour cell viability assay in hepatitis cells, and assess the mitochondrial toxicity by running cell viability assay in hepatitis cells cultured in medium containing either glucose or galactose. And the assays in DILI mechanistic panel can reveal possible reasons that a compound causes DILI. We offer a combination of 6 assays in this panel that assess the general toxicity in human primary hepatocytes, high-content imaging cytotoxicity assay that measures 5 cellular parameters, including cell number, cell membrane integrity, intracellular calcium level, nucleosides, as well as mitochondrial function. And cholestasis is one of the causes for DILI. Inhibition of DSAP, which is the major efflux pump for bile acid from hepatocytes, may lead to cholestasis. Nuclear receptor FXR activation may counteract the inhibition effect of DSAP by a compound. Therefore, by combination of DSAP inhibition assay with FXR antagonist assay, we can provide a better prediction on cholestasis. The induction of phospholipid OSIS and the glutathione conjugation assay are also included in the panel in order to cover a broad range of the causes of DILI. We use 2 types of cells in the assays that assess DILI risk: human primary hepatocytes and hepatitis cells. Those are there are advantages and disadvantages of each cell type. The human primary hepatocytes can be either freshly isolated or cryopreserved. With the improvement of cryopreservation technology, the viability and the characteristic of cryopreserved hepatocytes are relatively comparable to freshly isolated hepatocytes. They have the advantages of being more convenient, and we can conduct many experiments at different times from the same donor, which improves the reproducibility of the assay that use those cells. The human primary hepatocytes are considered as the gold standard for in vitro hepatotoxicity assay because they are the closest in vitro system to the in vivo condition. They return functional activity of major drug metabolizing enzymes and drug transporters under short-term 2D culture or relative longer-term in 3D culture or co-culture with other hepatic cells. But there are some limitations of using human primary hepatocytes, mainly due to phenotypical instability, short lifespan, batch-to-batch variation, which limits the reproducibility of the assay. The cells are significantly more expensive than the hepatitis cells, which also limits its usage in drug screening. The hepatitis cells are widely used in drug discovery for hepatotoxicity assessment. They are derived from human hepatoma cells. They have the advantages of high stability, unlimited lifespan, ready availability, and high reproducibility. The major limitations of hepatitis cells are that they generally do not express phase 1 and phase 2 drug metabolizing enzyme and drug transporters. It has limited usage for predicting the toxicity caused by metabolites of a drug or hydrophobic drugs that need hepatic transporters to get inside of the hepatocyte. And now I'm going to talk about the in vitro assays that assess mitochondrial toxicity. And drug-induced mitochondrial dysfunction is considered as one of the important determining factors in the onset of drug-induced liver injury. It is important to screen mitochondrial toxicity at early stage of drug discovery. Mitochondrial toxicity can be assessed by a set of cell viability assays in which hepatitis cells are cultured in the medium containing either glucose or galactose. When the tumor-derived cells, such as hepatitis cells, cultured in the medium rich in glucose, they generate energy by glycolysis, which bypass the mitochondria. It is the so-called Crabtree effect. They are resistant to many mitochondrial toxins. However, when the glucose is replaced with galactose, the cells are forced to use mitochondria to produce energy. Therefore, the cells cultured in galactose medium are more susceptible to mitochondrial toxins. It is commonly used in vitro assays to assess mitochondrial toxicity in drug discovery because it is simple, fast, high throughput, and more importantly, it has high predictive value. There are many peer-reviewed publications that validated the utility of this assay in identification of drug-caused mitochondrial toxicity. We have validated a high-throughput screening assay by using HepG2 cells captured in glucose versus galactose medium. In the validation study, we tested 20 reference compounds with 4 compounds that are non-mitochondrial toxins, highlighted in orange in the table here. The other compounds do not have mitochondrial toxicity but cause cell death, which are highlighted in gray, shown here, and 5 compounds that are not cytotoxic in this assay, which are highlighted in green. The assays can correctly distinguish the non-mitochondrial toxins from non-mitochondrial toxins by using the assay-50 ratio between glucose and galactose, which is presented in the last column of the table right here. We can see at the right-hand side of the slide the dramatic difference in those dose response curves from mitochondrial toxins. The green plots are the inhibition of cell viability from the cells captured in galactose medium, and the blue plots are from the glucose medium. The results demonstrate that the assay can be utilized to identify compounds that disrupt mitochondrial function by different mechanisms. For example, FCCP is shown in this plot. So the FCCP can uncouple mitochondrial oxidative phosphorylation from ATP synthesis, showed almost 20-fold shift of the assay-50 in this assay. Metalmycin C, the plot showing here, inhibits a complex 3 cytochrome C reductase in mitochondria, showed over 1,000-fold change of assay-50. Rotenone is a classic mitochondrial inhibitor that inhibits complex 1 NADH ubiquinol oxidoreductase in mitochondria. We observed over 100-fold of assay-50 shift between the glucose and the galactose assays. I want you to focus on the assay-50 ratio column in the table, which is the ratio between cell viability assay-50 obtained from cells captured in glucose medium versus galactose medium. The assay-50 ratio is below 3 for those non-mitochondrial toxins and much greater than 3 for those non-mitochondrial toxins. Assay-50 ratio gives us a clue that if a compound may cause mitochondrial toxicity, those assays can be run in either 96-well plate or 384-well plates with 5 to 10 business days turnaround time. Those assays can be utilized in early stage of drug discovery because they are predictive, requires minimum amount of compound, have fast turnaround time, and cost effective. We use high-content analysis imaging assays to illustrate the mechanisms of hepatotoxicity caused by test compounds. High-content analysis technology can analyze multiple parameters in the same cell simultaneously by staining the biomarkers with different fluorescent dyes. It provides rich information on cellular biology. This cartoon illustrates what happens when healthy cells are undergoing cytotoxic attack. In general, we will see the decrease of cell number due to cell death or inhibition of cell proliferation. We also may observe decrease in nucleosides, which is the result of chromatin condensation, is one of the hallmarks of apoptosis. Decrease in mitochondrial membrane potential, which indicates mitochondrial dysfunction. We also know that increase of intracellular calcium, membrane permeability, and reactive oxygen species may cause cytotoxicity. By measuring those cellular parameters, we can provide you with magnific information on the reasons that test compounds cause cytotoxicity and ultimately DILI. We offer a high-content imaging cytotoxicity panel in HepG2 cells, which measures 5 endpoints simultaneously in individual cells. In this assay, cell viability nuclear size, mitochondrial function, intracellular free calcium, and cell membrane integrity are visualized by the staining of different fluorescent dyes. As we shown here, the top panel of the images were taken from HepG2 cells that were treated with vehicle control. In this case, is 1% DMSO. The bottom panel of the images were taken from the HepG2 cells that were treated with 120 nanomolar cerevastatin. From the nuclear channel, which measures cell number and nuclear size, we can see that the number of cells is dramatically decreased after cells were treated with cerevastatin, and the nuclear size is also reduced. The functional mitochondria was visualized by the accumulation of a fluorescent dye inside the functional mitochondria, which is shown in yellow in this image. As they shown here, the intensity and the number of stained spots are significantly reduced in cerevastatin treated cells. The intracellular free calcium is shown as the bright spots in the image. We see a clear increase in both number and intensity of the stain, which indicates the increase of intracellular calcium. The interruption of cell membrane integrity is visualized by staining the cells with a membrane impermeable dye. If a cell is stained by the fluorescent dye, it indicates that the cell membrane is compromised. As is shown in this image, cerevastatin treated cells showed significant increase in the staining, indicating the damage of the cell membrane. This assay can be run at multiple concentrations to determine IC50 or EC50 of a test compound. After analyzing the images, those response curves can be plotted, and the IC50 and EC50 values can be calculated. As I'm showing in this slide, the effects of cerevastatin on nucleoside, mitochondrial membrane potential, intracellular free calcium, membrane permeability, and cell number are analyzed, and the IC50, EC50 values are reported. You can easily rank your compounds based on the IC50 or EC50 value of each measured parameters. We also use human primary hepatocytes in high-content analysis assay. As we can see in the image on the left, that the nuclei of human primary hepatocytes are stained by HERGs, showing in blue in the image, and the mitochondria are stained by TMRE dye and shown in green in this image. In the image on the right, the RS is stained by a fluorescent dye called H2D-FFDA, shown in red here. The blue staining in this image is the nuclear stain by HERGs. The results from the cells incubated 4 hours with amiodarone, which is an anti-arrhythmic drug, causes severe liver injury in clinic, are shown in this plot. The black trace is the cell viability plot. So we can see there is no significant cell death after 4-hour incubation. The blue trace is the induction of reactive oxygen species. We observed a significant and dose-dependent increase in ROS induction by amiodarone. The red trace here is from mitochondrial function, which is significantly decreased. This experiment demonstrated that the ROS and the mitochondrial function measurements are more sensitive than cell death, and they happen earlier than cell death. The plot here are from the cells treated with mitoxantrone after 24-hour incubation. The black trace is the cell viability, which shows dose-dependent cell death. The blue trace, which is for RS induction, is significantly increased, and the increase is dose-dependent. The red trace is from mitochondrial function, which is decreased in a dose-dependent manner in this experiment. And the utilization of high-content analysis imaging assays to predict DILI in humans has been extensively validated and there are a few publications of those validation studies. For example, in a study in 2006, O'Brien tested 243 compounds in hepatitis cells by high-content analysis imaging cytotoxicity assay. The results show 80% specificity and 93% sensitivity. Xu published his research in 2008, showing 95% specificity and 50% sensitivity by using human primary hepatocytes from over 300 compounds. In 2014, Garcett's research showed 75% specificity and 58% sensitivity using both human primary hepatocytes and hepatitis cells from 144 compounds. The results demonstrated that high-content analysis imaging cytotoxicity study can aid the in vitro detection of drugs that have a high propensity to cause DILI in vivo. If used to support compound profiling during drug discovery, such assays have the potential to aid selection of drugs with reduced likelihood to cause DILI in humans. The high-content analysis imaging assays that we offer are summarized in this slide. And those assays can be run in either hepatitis cells or human primary hepatocytes at any time point from 4 hours to 72 hours. It can measure multiple cellular parameters such as cell number, ROS, mitochondrial membrane potential, intracellular free calcium, nuclear size, membrane permeability, and heat shock protein 27 representing cellular stress. Those assays can be customized to meet your specific needs. Now I'm going to talk about the in vitro assays that we use to assess cholestasis. Cholestasis is one of the major causes of DILI. Clinically, cholestasis is defined as the reduction or stoppage of bile flow, which results in accumulation of bile acids and bile salts in hepatocytes. This cartoon shows the regulation of bile acid homeostasis in liver. The concentration of bile acids are regulated by multiple uptake and efflux transporters, including NTCP, which is responsible for the uptake of bile acids from blood to a hepatocyte, and efflux transporters, including BSEP, MRP2, 3, 4, and OST-AB, which are responsible for the clearance of bile acid from hepatocytes to blood or to a bile duct. And CYP7A1, which is the enzyme that catalyzes the bile acid synthesis in hepatocytes. This synchronized activity among transporters and enzymes keeps the bile acid in balance in healthy liver. If a drug blocks one of the bile acid efflux transporters, such as the BSEP, it may result in the accumulation of the bile acids in hepatocytes and lead to a cholestasis. Therefore, screening for BSEP inhibition has been proposed and used as one of the in vitro assays for DILI assessment. It turns out that it is not that simple. And there's a story of from two drugs that block BSEPs, triglycerol and pioglitazole. Both are type 2 diabetes drugs. Triglycerol was removed from market in 2000 due to DILI caused by cholestasis. Pioglitazole is still on market and considered as a safe drug to treat diabetes. Pioglitazole is a more potent BSEP blocker than triglycerol, as it is shown in this table that triglycerol inhibits BSEP with acidity of 2.7 micromolar, and pioglitazole has acidity of 0.3 micromolar. While triglycerol causes cholestasis, pioglitazole does not, even though it is a more potent BSEP blocker than triglycerol. As I mentioned in the last slide, the homeostasis of bile acid is regulated by transporters, which transport bile acid in and out of the hepatocytes, and CYP7A1, an enzyme that catalyzes bile acid synthesis. On top of those, The nuclear receptor FXR regulates the expression of those transporters and enzymes. Research found that bile acids serve as hormone-like signaling molecules to active nuclear receptor FXR, regulating homeostasis of themselves. When bile acid level increase in hepatocytes, which may be caused by the inhibition of B cells, the FXR is activated by the high concentration of bile acids. The activated FXR upregulates the expression of efflux transporters such as BSAP, MRP2, and OST, and downregulates the expression of the bile acid uptake transporters, NTCP, and CYP7A1, which is responsible for synthesis of bile acid in hepatocytes. So this way, the homeostasis of bile acid is maintained, even though the clearance of bile acid by BSAP is blocked. Inhibition of BSAP alone may not cause cholestasis. It is discovered that triglycerol is also an antagonist of FXR. Therefore, it inhibits not only BSAP, but also the feedback compensation regulatory pathway. Pyroglycerol is not an antagonist of FXR. This explains why triglycerol causes cholestasis. Moreover, a research by Chen in 2018 showed that the correlation between BSAP inhibition and DILI outcome is not significant. BSAP inhibition alone may not be predictive for cholestasis. Therefore, by combining the screening of BSAP with FXR antagonist and agonist screening will help improve the productivity of the cholestasis. We have validated the BSAP inhibition and FXR antagonist and agonist assay at Eurofins Discovery. We use inside-out vesicles from HEK cells that overexpress in human BSAP. In this assay, a tritium-labeled taurocholate is used as substrate. Based on a set of reference compounds, the results of BSAP inhibition assay in our lab are comparable with the values from literature. By combining the BSAP inhibition and FXR agonist and antagonist assay, we can provide a better assessment of cholestasis. This is the summary of the in vitro assays that we use to assess DILI risk at Eurofins Discovery. We offer two panels of assays, DILI screening panel and the DILI mechanistic panel. The assays in DILI screening panel are high throughput, low cost, fast turnaround assays that can be used at early stage of drug discovery to screen large amounts of compounds. In this panel, we use HEP2 cells to assess cell viability after 72-hour incubation. And we use two cell viability assays to assess mitochondrial toxicity. We offer the combination of 6 assays in DILI and a mechanistic panel in order to provide mechanistic information on the causes of DILI. Those assays include high content analysis imaging cytotoxicity assay in HEP3 cells or human primary hepatocytes, BSAP inhibition, and FXR agonist and antagonist assay that assess cholestasis, also phospholipidosis and glutathione conjugation assays. By offering the two panels of assays, we can provide our clients with a useful tool to de-risk their drug candidates' DILI liability at early stage of drug discovery. I will take a few minutes to give you a brief overview of Eurofins Discovery and admin toxicology services at Eurofins Discovery. Eurofins Scientific is an international life science company. It provides services in food, environment, pharmaceutical products testing, and drug discovery CRO services. Eurofins Discovery is the Eurofins Drug Discovery CRO services branch. There are more than 600 scientists working at Eurofins Discovery, with 7 locations worldwide located in San Diego, San Francisco in California, St. Louis in Missouri, France, Spain, and the UK in Europe, and Taipei in Asia. And by combining the market-leading drug discovery expertise and capability, Eurofin Discovery strives to provide the solution for every problem you have in your drug discovery program, from compound synthesis, admin toxicology, to in vitro safety assessment. We have been providing admin toxicology services in drug discovery for over 20 years. Eurofin Discovery has extensive experience in consulting, designing, and conducting in vitro admin and toxicology testing programs for thousands of clients in the pharmaceutical, biotech, consumer health, as well as for leading academia and government institutions. We offer over 450 in vitro admin and toxicology assays, which provide you with a comprehensive solution for assessing drug bioavailability, metabolism, drug-drug interaction, toxicity, and safety. Our scientists have many years of industry expertise in both DMPK and toxicology. We provide reproducible data with fast turnaround times. Our services are flexible in order to provide customized solutions to fit your testing needs. We offer a comprehensive panel of in vitro toxicology assays to de-risk our drug candidates at early stage of drug discovery program. Besides of the assays that assess hepatotoxicity that I discussed today, we also offer genotoxicity, cardiac safety, renal toxicity, and cytotoxicity assays. And those in vitro toxicology assays can be utilized at the early stage of the drug discovery to de-risk your drug candidates, from hit to lead optimization to candidate selection. And this concludes my presentation. I'll be happy to answer any questions that you have. Thank you for your time.
Great. Thank you, Dr. Zhao, for an excellent presentation. It looks like a few of you have already submitted questions while our presenters are answering your questions. Please take a moment to answer the feedback form that appears in the bottom of your screen. All right, so let's get to the Q&A. So the first question that came in, how do you choose which type of cells, my apologies, which type of cells to use, HepG2 cells or human primary hepatocytes in DILI assessments?
Thank you for the question. So both of the cells are widely used for hepatotoxicity screening and drug discovery. As I mentioned in my presentation, the human primary hepatocytes are gold standard in vitro hepatotoxicity testing because they express the major drug metabolism enzymes, including phase 1 and phase 2 enzymes and drug transporters in short-term 2D culture and relatively longer-term in 3D culture or co-culture with other hepatic cells. There are publications show that the major CYP enzymes maintain stable level after 10 to 15 days or even longer in 3D culture. So the human primary hepatocytes are suitable to measure long-term effect in the 3D culture to simulate the in vivo condition. However, the enzyme expression and activity profiles are donor dependent, and the total amount of the cells from a single donor is limited, which drastically reduce the reproducibility of the assay if you use the cells from different donors. It is also more expensive than the HepG2 cells. On the other hand, the HepG2 cells are tumor-derived cells which have unlimited availability. It is convenient, and the assays using HepG2 cells are more reproducible and cost-effective. However, they do not express the major drug metabolism enzymes and drug transporters, which limits its usage for assessing the toxicity of the metabolites of the drug and the drugs that need drug transporters to get inside of the cells. Based on those pros and cons of each cell type, I would recommend using HepG2 cells in initial screening assays to screen large amounts of compounds because the assays using HepG2 cells are simple, fast, cost-effective, and using the human primary hepatocytes to confirm the negative results that's from the lead compound.
Awesome. Thank you for that. The next question that has come in, can you talk about phospholipid OSIS assays and how it applies to DILI assessment?
And thanks for the question. So I apologize that I did not get into the details of this assay in my presentation due to the time limit. Their reports show that some DILI compounds can cause steatosis and phospholipidosis, such as valproate and amylaldehyde. Screening phospholipidosis in vitro can provide useful information of DILI assessment. We know that the cationic antiphilic drugs, such as antibiotics, antidepressants, antihistamines, and some other prescription drugs, have been identified as inducers of phospholipidosis in animal and cell culture model. The drug-induced phospholipidosis is characterized by the appearance of intracellular phospholipids in lysosomes, and detection of drug-induced phospholipidosis have been performed using electron microscopy, which is a time-consuming and labor-intensive technique. More recently, fluorescent dyes have been employed to assess phospholipidoses in a high-throughput manner. We use a high-throughput assay using a commercial available fluorescent dye called lipid tox. This dye is a fluorescent lipophilic dye. It has been shown that it's highly sensitive in detecting drug-induced phospholipidosis in HepG2 cells. And this fluorescent probe methodology can be performed rapidly and requires only a small amount of compound. We offer a rapid high-content analysis assay to assess drug-induced phospholipidosis in HepG2 cells after 48-hour incubation. And so this assay is fully validated by a set of reference compounds and robust and reproducible. And the steatosis can also be evaluated at the same time with a different fluorescent dye in this assay.
Great, thank you. The next question that has come in, what are the most predictive cellular measurements in the high-content imaging assays in terms of in vitro DILI assessments?
The high-content analysis is a very powerful tool to evaluate cellular response to a cytotoxic compound in individual cells. It can measure many of the different cellular endpoints that reflect the cytotoxic effect simultaneously, such as the mitochondrial dysfunction, cell membrane integrity, intracellular free calcium level, and caspase-3 expression, which indicates the induction of apoptosis, and heat shock protein expression, which indicates the cellular stress. We can measure almost any proteins which has a cytotoxic indication as long as there is available antibody. However, it is an information-rich assay. So in order to determine which assay or which endpoints are more relevant in predicting DILI, we collaborate with AstraZeneca in 2014. We tested a total of 144 compounds, and 108 of them that's a non-DILI compound. So we use high-content imaging and assay in both HepG2 cells and human primary hepatocytes. So we test those compounds with the incubation time of 4, 24, and 48 hours, and measured multiple endpoints, including the mitochondrial function, the ROS, heat shock protein, and phosphorylated histone H3, as well as phospholipidosis. And the result of this study was published in Toxicology in Vitro in 2014. So we found that the individual endpoints which identified drugs that cause DILI with greatest precision was the maximal fold induction of RS, reactive oxygen species, in human primary hepatocytes after 24-hour incubation. This is one of the conclusions we got from that study. I guess so you also need to consider the chemical property of your compound and which class it belongs to determine which parameter is more relevant in prediction of DILI.
Awesome, thank you for that. And we have one more question. Do you suggest to use endogenous substrates in the BSEP inhibition assay?
Yes. So using endogenous substrates can avoid the problem of using artificial substrates, such as multiple binding sites on the transporters or different affinity of the substrates on the transporters. So there are reports that there are multiple binding sites of substrates on certain transporters. If the binding site of the artificial substrates is different from the endogenous substrates, it may give a different result in the inhibition assay when you use a non-endogenous substrate. So using endogenous substrates provides the closest result to the in vivo condition. So in our assay, we use a tritium labeled taurocolic as the substrate for this assay. So there are reports that some other fluorescent labeled taurocolic can be used as substrates to simplify the experiment. The assays that use non-endogenous substrates in the B-side inhibition assay need to be fully validated before being used in the screening. So, so in our lab, we are actively explore these methods as well.
Awesome, thank you for that. Well, I'm afraid we have run out of time. I'd like to thank Yang for an outstanding discussion. If we haven't answered your questions, we will follow up after the webinar. On behalf of Xconomy, have a productive remainder of the day.